Hello, everyone, and welcome to the Tree of Sex session at Biodiversity Genomics 24. Um, Let me open with a very short introduction to the consortium, and then we will continue with three contributed talks. So exciting. And we will finish our session with a short Q&A session. So the Tree of Sex initiative is an organization that aims to that aggregates a large body of reproductive biologists across the whole globe. We are more than 150 at the moment and from more than 20 countries. And as you see on these word clouds, these are the topics that we focus on. So we really focus on lots of different things. It's not just we're not specialized in individual systems. We are basically trying to generate this very wide array of knowledge across the types of reproduction, different aspects of reproduction, as well as different systems. And the reason why we do that is because our main aim is to create, integrate knowledge relevant to the evolution of reproductive systems in eukaryotes in a scalable, accessible, and flexible database. So what we would like to do is to just generate this one database where you will be able to search all the reproductive knowledge for all the species of eukaryotes. And on the way there, we're just creating this large global collaborative network of reproductive biologists that already is proving to be extremely useful. So while the point one is just a work in progress, the point number two is actually already happening. And right now, what we're doing to make the first goal happen is that we're organized in 6 teams that are focusing on different aspects. On the ontology team is trying to find how we can harmonize vocabularies from all the systems in a meaningful way. We're trying to search the literature that would need to be integrated, ethics teams that are considering all the problematic questions of our choices. Of course, the funding team as well as database integration team, because we're not the first reproductive database out there, and communications team. And so this is how we're right now. We're in the operation regime because we're trying to kick off the database, but we're also soon going to be launching all sorts of research topic teams that will be across the consortia focusing on the different research questions. So to get everybody on board, we organize annual workshops where we discuss what we're doing, how we're doing, and what to do next. And we have also very cool shirts. That's actually not how they look, but and the next workshop will be in Barcelona next year, so maybe hope to see you there. And the main reason why I'm here pitching you of what we're doing is because we're an open consortium. I hope that you will be part of it too. So join us. Everybody's welcome. If you're not sure how you can contribute, I would like to reassure you that there are many, many ways how you can. That right now, it's all around on volunteer efforts, on sorting out the ontologies and creating data lists of databases and trying to figure out how they could be put together. So it's lots of work, and but the community is very enthusiastic, and we are a great bunch. I'm sure you will have a great time with us. So if you just go to our webpage and there is a register button, if it's not there right now, just wait for a few hours. But there will be a register button in the right top corner, and if you just click on it, you will be able to log in with your ORCID account, so you don't have to have a new password or anything, and you will just give us a bit of your details about yourself, and you will be automatically integrated at all the mailing lists and part of the consortium. So with this, I would like to proceed to our beautiful talks.
Hello, so my name is Dr. Chloe Vanderberg, and I am a postdoc at the University of Otago in New Zealand. And today I'm going to be talking about the New Zealand spotty wrasse as a model for sexual fate genomics. So in this talk today, I'm going to go a little bit about the background on spotty behavior and sex phenotypes, talk about the social dynamics at the neurobehavioral level, talk a little bit about the gonadal gene expression throughout the sex change, and if I've got some time at the end, talk a little bit about some future work that we'd like to do in an ideal world. So, the New Zealand spotty wrasse is a fish that is found ubiquitously throughout the New Zealand coastline. You can see it pretty much anywhere where you go out diving. There's a couple of photos here from some friends that took some photos for me of them out in the harbour in Dunedin, where I live. And so the spotty is a proteigenous sequential hermaphrodite, so it changes sex from female to male. So they have these really plastic sex systems, and they also have so they have these juveniles, which then can differentiate into these initial phase females or these initial phase males, which externally look super similar. You cannot tell them apart. And what I'm mostly focusing on is this the sex change that happens from the initial phase or IP female to TP male, the terminal phase male, and you can also see there's a bit of a coloration change that happens too. But these IP males can also actually role change and become TP males as well. So, thing to know about spotties is that their sex change is socially cued. It takes approximately 60 days for them to change sex, which is quite slow. It occurs outside of the breeding season, and so the sex change is a complete functional conversion of the gonads from the female to male. So you can see some images of some histology pictures here of these females converting all the way through early transitioning, mid transitioning, late transitioning, and then into a TP male gonad. So the thing to know about the socially cued sex change is that it's all about dominance. So the most dominant female will be the one that changes sex. So if the TP male is removed for whatever reason, whichever female in that sort of social group is going to be one that changes sex. So we are doing a lot of work at the moment looking at the different sort of behavior of these groups of the females to try and understand how they know which one is the most dominant one. And a lot of this work's being led by the PhD student Hayley Katermis, and we've got some of this work currently in review at the moment. So we did an experiment where we set up multiple tanks with 5 fish of all different sizes, 5 female fish, and we found this really clear dominance hierarchy based based almost entirely on size. And so when we talk about dominance, there's a few different behaviors we looked at, but some of the main ones are these rush behaviors and these escape behaviors. So we found, there's a really clear trend of the biggest fish will do the most rushing, and the smallest fish will do the most escaping. And you can see a little picture here of the most dominant fish. They like to hide out in these little PVC pipes, and you can see there's a fish that came along and tried to check out the other fish in the PVC pipe, and it chased it away. So you can sort of see a few stills there. Cool. So after we set up those tanks, what we then did was we wanted to see what happened when we disrupted the social hierarchy. So we had the fish in these stable groups for 2 weeks, and then we removed the rank 1 fish, the biggest one, for an hour, and we found straight away that the behavior of the rank 2 fish became extremely aggressive very quickly. We also found, which you could see in the I don't know if you can see my mouse wriggling, but that's the dark red bar in the rank 2 of the graph. We saw this sort of trend through the other ranks as well, although it wasn't actually significant for those. So then we sampled the fish. So we wanted to see what was going on in their brains. So we looked at the neural activation of the mesolimbic reward system and the social decision-making network, and use this PS6 antibody to stain to look for neural activation. We found that the rank 2 fish showed significant differences in many of these different regions in the brain. So we found that submissive behaviors such as escaping and swimming was quite correlated with highly correlated with the ATN region, which you can see in the graph at the bottom left, that in rank 2 fish, this was the only one where it was a lower neural activation to the other fish. And we found many different aggressive behaviors were correlated with many different regions of the brain. There's a couple there as an example. But in the rank 2 fish, the neural activation number of cells were much higher than all the other fish. So what does all this mean? Why do we want to understand spotty social hierarchy? So what we find is that the fish that's next in line to the throne, it already knows its place in the hierarchy. This is quite important. So, and we know that establishing dominance is the initial step before sex change occurs, which is really important for us to be able to see behaviorally as externally, it's almost impossible to tell when a fish has begun to change sexes. They do start to have these coloration changes to become TP male, but in the first sort of initial time when it's happening, which starts happening very early, even though it takes 60 days for them to change sex, we see these changes start occurring even an hour after a disruption to their social hierarchy. So I'm going to change gears a little bit and talk about this gonad transcriptome dataset that I've been working on at the moment. So I won't go into too much of the details of the different studies that it came from, but there was 4 different experiments that occurred in our lab over the years. So just shout out to those people because this was not data I generated, so quite a lot of people went into generating this data. But essentially, a lot of this data covers a sex change data set. So they generated samples for fish that were transitioning from female all the way through early transitioning, mid transitioning, late transitioning, and terminal phase males, as well as collecting some of these IP males as well. So what I find is that these samples separate primarily based on gonad histology from female to male. So you can see the right, this PCA, the female in the sort of pink and purple colors are clustering together, and the males on the left, the TP males in blue and the in gold. So the IP males are more on the left here, but these the transitioning samples are bridging bridging through the PCA. I find that there's unique signatures of gene expression for each of these states. So these graphs are showing the top 30 genes that are correlated with each state and what their expression looks like across each transitional state. So these are the ones most positively correlated, and you can see that for each state. Some are stronger than others, but there is, yeah, an increase in expression. There's genes that typify each of these states. This is probably a good point to point out again that I am mostly looking at the sex change. So it sort of looks like in these graphs, like they move through like mid transitioning, late transitioning, and then to IP male to terminal phase male. That's not what happens. That I've just included these IP males next to TP males, so it's something to have a look at as well. But they're not transitioning through the IP male state. So in the interest of time, I'm not going to get too much into all the different like gene ontology terms and stuff that I found. But what I find is that females are associated with increase in expression of things such as phosphatidylainositol and lipid signaling, and a lot of different transcription stuff happening, and decreased expression in things such as meiosis, cell regulation, immune system. I think some of this meiosis stuff might sound slightly surprising at first. It has decreased. I think that's because these samples are non-breeding and breeding females as well. And so if you're into sort of the sex change literature, you'll know that there's often a sort of classic set of sex change genes people tend to always investigate. And so what I find looking at these genes, it's just an example of a couple of them, is that they tend to be expressed somewhat higher through to mid-transitioning state before they start dropping off, with the exception of aromatase, which is known to be that early or likely an early initiator of sex change. But I did identify, and this is just an example of 3 of them, of there are quite a few genes that are high in females and early transitioning, but drop off really quickly in the other samples. So these are ones that in my quick look of the literature, I couldn't find that they were linked to sex change or sex determination. But yeah, it's quite cool to know there's potentially quite a few novel targets that I'm still looking into. So IP males are associated with increased expression in things like ciliary assembly function, protein organelle turnover, energy metabolism, decreased expression in neuronal and synaptic function, actin cytoskeletal muscle development, stuff like that. But what's most interesting, I think, as well with these IP males is that they have these really unique patterns of highly expressed and lowly expressed genes that are quite different to TP males. So these genes are ones that are highly correlated with the IP male state, but it's very interesting to me that even though both these are gonadal samples, but with spermatogonial cells from IP males and TP males, but they're really different expression of like a lot of different genes, which I think is very interesting. And so TP males are associated with this increased expression in glycogen lipid metabolism, ECM remodeling, protein processing, and decreased expression in things such as reproduction, sperm-egg interaction. I think this is possibly because this reproduction thing, that might also be because these are terms that are very high in female samples, this reproduction sort of stuff. And that's why they're actually very low in males, is my guess. And possibly good to note as well that, yeah, because these TP males came from data sets that were intentionally inducing sex change, these are fish that have more recently changed sex to become a TP male, which I think is why you see some really high like energy metabolism stuff going on too. And there's a huge change that happens with the just the tissue remodeling and stuff as well. So genes that are highly correlated with TP males tend to start being expressed quite highly from once we hit about that late transitioning stage. So there seems to be quite a demarcation from that sort of mid-transitioning to late transitioning stage that seems to be happening with a lot of genes. But frequently these genes are also expressed higher in IP males than TP males. So when I was looking at which genes were highly correlated with the TP male trait, there were not many that were not also even more highly expressed in IP males, which I find very interesting. So my sort of conclusion was that IP males are very unique, but TP males are not super unique. They're actually a bit more similar to IP males in some way, if that makes any sense. And so, yeah, transitional states are associated with increased expression in things such as transcription for early transitioning, transcription, protein synthesis, modification, degradation, cell proliferation. A lot of these early transitioning samples were very similar in expression to females, which makes sense here because we're not actually seeing a huge difference in their expression. The mid-transitioning state was probably one of the ones that popped up with one of the most metabolically active state with a lot of terms for rapid growth and differentiation, protein remodeling, fatty acid metabolism. Late transitioning is where we really start to see a bit more of a male-like profile. This increase in steroidogenesis. The TLR pathway popped up a lot, which I thought was also a bit interesting because the females tend to have this more decreased, more of a decreased immune genes, and then in the males they had these more increased immune genes. So that's something potentially to look at as well. Definitely a lot of structural changes popped up in this tissue, which again made sense because that's really a lot of structural changes happening in the gonad. And sort of overall what we're seeing is that transitioning is characterized by this first increase in these a lot of intracellular processes, followed by a very metabolically active state with rapid growth and differentiation, and then substantial energy going into new tissue formation and steroid hormone biosynthesis. So some conclusions, changes in neural activation and dominance/submissive behavior are strongly linked to fish rank. IP males and TP males have vastly different gonadal gene expression profiles, despite both being functionally males, both being able to produce sperm and which could fertilize eggs. There's very specific transcriptional profiles that are associated with each stage of sex change, which show how the tissue become increasingly more metabolically active, prioritizing cell differentiation, growth, and structural changes at different stages of sex change. And all of these sorts of things is why I think, well, we know spotties have very highly plastic sex systems, and that's why we think they're really great models for understanding the complexities of sexual fate genomics. And so some stuff that we're currently doing / future work is looking at generating a single cell data set for that early gene expression that happens in the brain post the social disruption. So now that we understand the behavior and the sort of neural activation in their brains 1 hour after we've disrupted their hierarchies, I'm working on this data set at the moment, having some lab troubles, but hope we can still get that up to have a look at just single cell sequencing of the brain, see what's going on in there and get that really in-depth insight. Further untangling the dynamics of the hierarchy disruption and how female spotties can be primed to change sex. So that's we're doing a bunch of more experiments October, November around this to get an even better idea of the sort of behavioral stuff that's going on. Of course, some more molecular sampling. In future, I'd really like to have a look at this idea of investigating the juvenile sex determination. So those juveniles that can become IP males and IP females, if we can manipulate that. I should probably mention that, we have a genome for these fish, but we don't have, they don't have sex chromosomes that we've found, and we don't really know how they actually determine what their sex is. So it'd be really cool to see what sort of social and environmental factors we could do to figure out how they're deciding which fish are changing sex or becoming IP males, IP females. Lots of acknowledgements. There's an amazing group of people that are working on what we call the sexy fish team. And yeah, a lot of this work was, I mean, we couldn't have done it. Was a lot of this work is not just me, it's a lot of other people as well, and some people in the lab as well that have in the past contributed to a lot of the data generation, the sampling, a lot of just fish logistics. So thank you to everyone for that. And thanks everyone for watching.
Hello everyone, my name is Arif. I'm a second year PhD student in the Tree of Life program at the Wellcome Sanger Institute, University of Cambridge. Thank you first for including my work in the Tree of Sex session at the BG24, in which I will be presenting on the sex chromosome evolution in Coleoptera, or more broadly, the chromosomal evolution of beetles. J.B.S. Haldane, in his book What is Life, published in the 1940s, said that the creator would appear as endowed with a passion for stars on the one hand and for beetles on the other. So this is of course very reasonable, simply because there are nearly 400,000 species of them, and perhaps more. So comparing that, for example, to mammals or birds would make them appear super tiny, because in fact, only 3% of them compared to beetles. And of course, it has gained our curiosity since the early development of biology. We know that Darwin had a deep fascination with beetles, as did Wallace as well, who collected around 83,000 specimens of beetles, which roughly around 60% of what he collected during his trip in Southeast Asia between 1854 and 1862. So, to me, there are countless reasons as to why we should study them, and for someone who's interested in speciation, they provide us with an arena to solve evolutionary problems. Originating in the Carboniferous era, beetles are considered an example of ancient radiation. So co-diversifying with angiosperms, herbivory or plant-feeding lifestyle is thought to be the key to their diversification, evolving multiple times and facilitated by horizontal gene transfer that allows them to digest plant cell walls. Several other studies have also explored the pattern of genomic architecture in beetles. For example, in 2021, the first chromosome scale genome for Phytophaga from a species of Pachyranchus was sequenced. So by comparing it to other saffron beetle genomes, they revealed that chromosomal syntenic patterns were almost absent in beetles, with gene order thoroughly shuffled along the chromosomes. Or more recently, last year, it was suggested that despite the strong intrachromosomal rearrangement, their identity is somewhat conserved, and it was proposed that there are 9 ancestral linkage groups, which they termed as Stevens elements, named after Nettie Stevens, the American geneticist whose pioneering work using the beetle Tenebrium molitor helped identify sex chromosomes in the early 1900s. However, there are no large-scale genomic studies yet to completely resolve the issue. The shortcoming of previous studies is that the authors not only mainly assess syntenic blocks visually, but also they exclude Adivaga, which is ground beetles and other beetles. And we know that this group is crucial since they exhibit variation in chromosomal number. Meanwhile, 70% of the available beetle genomes were sequenced by the Sanger Institute, with which I'm affiliated, and almost 3 times as many are still in progress. So the idea is, can we do something with this bulk of data, which now accounts for 183 genomes? So what you're looking at here is the phylogeny of those genomes derived from the Busco genes, 177 species of beetles with far outgroups from Strepsiptera, Neuroptera, Megaloptera, and Rhaphidioptera. So if you see here, the spread of the species is quite nice. with representatives of important superfamilies, about maybe 60% out of 27 superfamilies. We don't actually have Archostemata and Myxophaga yet, which form the sister group to Adiphaga. And at the bottom are the genome size and the chromosome number, with alternating colors marking different superfamilies. The genome size can vary from half gigabase to 2 gigabase, with chromosome numbers ranging from 5 to 39. And there is no clear correlation between the two. Such variation suggests something, that while the chromosomal numbers are mostly similar among lineages, in a few there must have been some sort of rearrangement events. So our assessment using SynGraft inferred that this is indeed the case, with rampant fusions and fissions scattered throughout the phylogeny, in rough beetles, in weevils, in leaf beetles, and ground beetles, and so on and so on. So the rest have few rearrangements, even in some of the deeper branches. The number of fissions events is shown by coloring the branches, and the little dots in different colors indicate different sex determination systems. In terms of ancestral linkage groups, we inferred that beetles have 8 ancestral chromosomes, one less than the previously proposed ancestral linkage groups. So I made this simplified version of ancestral linkage groups for 6 species to make it easier to illustrate why. So if you look at S1, shown in turquoise, it splits into 2. And for many species in Polyphaga, this holds true, but not for all. And this split is what was detected in the previous ancestral linkage group proposal, and is almost identical for many species in the group. Meanwhile, in Adiphaga, instances where S1 splits into 2 are virtually never observed. So to me, it is clear that this is the main reason why they fail to infer the ancestral linkage group as 8. When you look at the synteny, you can see that S1 is split into multiple chromosomes, and the set of genes in each of these fission chromosomes is different for each species, despite having the same identity. We tested this by removing this clade, which, as expected, resulted in 9 ancestral linkage groups. So it is, to me, thus very clear that an ancient fission involving S1 occurred on the branch leading to Polyphaga. So such a robust chromosomal identity is extremely well preserved in many beetle lineages, even at the deeper branches. This is shown here as a synteny plot, with the size of the plot corresponding to the genome size. As you can see, while very little has changed in the number of chromosomes, we observe genome size expansion in some lineages, while in others it has not. Here, once again, I illustrate how the identity of each chromosome is largely conserved, with only a few simple fusions and fissions across the large scale of the phylogeny. Similarly, the sex chromosome, marked in pink, is also most of the time highly conserved. In Chrysomelidae and many other groups, however, things are a bit different. Many species underwent fusions and fissions, with some even splitting into nearly 40 chromosomes. So these highly rearranged lineages evolved independently, as their sister lineages seem to have quite conserved synteny. Interestingly, if you remember from the beginning, this coincides with the event that marked the birth of diversification in beetle evolution. Although many of these rearrangements events occur mostly at the tips, the inferred chromosome number at the nodes is mainly conserved between 9 and 10. In regard of the sex chromosome or the X chromosome, as mentioned briefly, it is mainly conserved, with several species undergoing independent loss of the Y chromosomes, resulting in an XO system. Some species, however, acquire additional elements in their X forming the neo-XY or the neo-X0 system. Shown here in pink are the X chromosomes of these 6 selected species across the phylogeny with autosome and X fusion. And this beautifully illustrates how the formation of the neo-X, despite involving the same type of X autosome fusion, results in different sets of genes with varying gene orders. Here on the left is the heat map showing how conserved the gene order is within the X chromosome across the different species and clades. And you can see that despite the conserved identity, their synteny is mostly conserved only within the same genus or within the same families, with very few rearrangement events. It was also suggested in the brilliant Lepidoptera chromosomal evolution paper by Charlotte Wright and others earlier this year that there is a clear association between the size of the elements and the number of rearrangement events, and that some elements are more likely to fuse with certain other elements. But here we see that it seems equally likely for each element to pair, but the number of events is not equally similar. This is probably related to how each element has a similar number of markers and the proportional length of the chromosome in which they reside. And I would like to finish by saying a little bit about transposable elements. At the moment, I'm trying to get those repeats annotated, and so far I've managed to annotate half of them. We know that there is a new result posted on bioRxiv in the middle of this year on how TEs rearrange structure variants, particularly inversions, and how this is associated with adaptive functions among different subspecies of deer mice in North America. And I think it is equally interesting to ask similar questions in beetles. How often, for instance, are ecological shifts associated with SVs or rearrangements? And what is the role of Ts in this? Can we identify patterns where specific types of Ts drive specific types of SVs and rearrangements? And why do we see genome expansions with little chromosomal reshuffling? And on the other hand, no expansions with rampant chromosomal rearrangements? And there are many other exciting questions to explore that will eventually allow us to say more about their role in beetle speciation and radiation, and also why Darwin and Wallace were particularly fond of beetles. So thank you very much. And if you have any questions, you can send me a message by email.
Hi, my name is Meredith Dolman, and I'm a postdoc at the Field Museum in Chicago. And today I'll be talking about my project on rethinking asexuality, sexual genes in Lepraria lichens. Evolutionary biologists have long asked why sexual reproduction is so common and whether long-term asexuality is really possible. Sexual reproduction is fairly costly energetically, but it comes with the benefits of creating new trait combinations for selection to act on. On the other hand, asexuality can be very beneficial because it maintains beneficial traits in constant environments. But on the other hand, it comes with a lot of downsides, including inefficient selection, which can lead to long-term deleterious consequences. There are, and these are on the screen, some supposed ancient asexuals, which seem to contradict the idea that asexuality is in general a dead end. But the more genomic resources that we collect on these and other systems, the more it appears that some sort of sex is often occurring and that there are very few truly long-term asexual lineages. In my group at the Field Museum, we work on lichens, which are a really beautiful and diverse system in which to study this question. And they have history of many transitions between sexual and asexual reproduction. As a means of introduction to lichens, they are a symbiosis composed of a fungal component called the mycobiont and the photobiont, which is an algal or cyanobacterial component. And together they form the lichen thallus, which is a whole new morphology that looks different from either one of the components. And this symbiosis is critical to the existence of the lichen. Now, sexual reproduction in lichens is pretty obvious. They form these apothecia that release ascospores into the environment, which are the product of sexual reproduction. Sexual reproduction in lichens is pretty complicated, but the important part is the sexual reproduction only occurs with the fungal genome, and these ascospores are only the fungal genome. So when they settle in the environment, they have to regain their algal symbiont, which could be a cost, an additional cost to sexuality that is not present in other systems. So why not take your photobiont with you? This is what asexual lichens do. They form these small granules of thallus, which include both the mycobiont and the photobiont, called ceridia, and these bud off and go settle in the environment, taking both components of the symbiosis with them, which could be an added benefit, but still comes with all of the traditional costs of asexuality. There are, however, some lineages of lichens, such as the genus Lepraria, which seem to be obligately asexual. They have this kind of powdery morphology. They have never been observed to form sexual structures, and their whole morphology seems to be composed of, look like ceridia, look like these little budding asexual reproductive structures. These are, however, a pretty old lineage. They're about 30 million years old, and there's a pretty incredible amount of diversity within this supposedly completely asexual lineage, which kind of goes against the idea that asexual lineages may be dead ends. For us, it's really convenient that this entirely asexual lineage is sister to a sexual lineage, the genus Stereocolon, which produces really obvious apothecia and sexual reproductive organs. So this allows us to take a genomic approach, use comparative genomics to ask this major question. Do Lepraria genomes suggest that this lineage has lost the ability to have sex? We took an approach using 3 lines of evidence. We first asked, does the Lepraria genome show evidence of a non-functional mating system? Then we asked, does it show evidence of loss of function in some core meiosis genes and other genes involved in the formation of sexual reproductive structures? And finally, we asked, is there a pattern in general of relaxed selection on these genes involved in sexual reproduction? So first, we address the mating type locus. Instead of having sex chromosomes like, typical vertebrates would, fungi and the fungi that are in the lichen symbiosis have a mat locus, a mating locus, which includes a primary mating type gene, and at least one auxiliary gene. In order for sexual reproduction to happen, 2 different mating type genes have to come in contact with each other. These alternative mating type haplotypes are in fungi known as idiomorphs. So 2 idiomorphs are needed, 2 alternate idiomorphs are needed for sexual reproduction to occur. They can either both be present in the same genome, which is known as a homothallic genome, and these individuals can self. However, many lichens, including the vast majority of Lecanoromycetes, which is the class that includes Stereocolon and Lepraria, are what is known as heterothallic. They have a single copy of either one or the other idiomorph in their genome, and they have to outcross. They have to mate with an individual that has the alternative idiomorph. So in our study, we first started with Stereocolon as the null model because it's sexual, so we can culture the fungus by itself. And since it's sexual, we assume that all of these genes are functional in Stereocolon. We assembled a high quality nanopore genome and pulled out the mating type locus. We found 1 mating type locus, indicating that it is heterothallic, like we would expect for Lecanoromycetes. And we did a conserved domain search and found the expected alpha domain in this MAT111 gene, indicating that it functions like other Lecanoromycetes and like we would expect. Lepraria, on the other hand, the supposed asexual, doesn't produce ascospores, so we can't culture the fungus on its own. So we had to do an Oxford Nanopore sequencing of the whole metagenome and then pull out the fungal components based on comparison with Stereocolon. Again, we found that Lepraria appears to be heterophilic. It has one copy of the mating type locus, and the mating type gene has the expected HMG box functional domain. So it appears functional, has the same start and stop codons that we find in stereocolon, which I'll show you in a second. So appears functional. In this study, we also took a phylogenetic approach. We sequenced an additional about 10 short read genome metagenomes from each genus, and then looked at their mating type locus. And stereocolon had both idiomorphs, and in each genome we found one copy of the idiomorph. So it is heterophilic as expected. Lepraria, if it was truly asexual for its entire or most of the history of this lineage, we might expect that this mating type locus isn't in use. And if it's not lost, and we know it's not lost, we might expect it to fix for one or the other of the idiomorphs if both aren't necessary for sexual reproduction. However, we find both idiomorphs segregating across the genus. And importantly, in this Lepraria finki, where we have two individuals sequenced, we have both alternate idiomorphs, suggesting that both are segregating in the population, and therefore suggesting that the mating type locus is probably functional in Lepraria. And this is the same thing we see when we have two individuals of the same genus in stereocolon, the sexual genus, as we would expect. So if we go back to our 3 lines of evidence, our first question was, does Lepraria have non-functional mating systems? And the answer is probably no. Its mating system seems functional, and seems to be in line with other Lecanoromycetes, many of which are sexual. Next, we asked about function of core meiosis genes, and we started by looking at there are 29 genes that are part of a bioinformatic toolkit for assessing reproductive mode in organisms based on their genome sequence. Here, I'll focus on 9 of these, which are thought to be meiosis specific and not have a role in any other functions except for during meiosis. Again, so I'll just go through our workflow using REC8, which is one of these meiosis specific genes. Again, we started with stereocolon as the null model because we knew we had only the fungal genome, and we knew that this is sexual, and so it should have functional meiosis genes. We pulled out the target gene, which here is REC8, and the 2 flanking genes, and made sure that they were indeed fungal, which we expect because this is a fungal culture. We then checked the start and stop codons of the gene model and compared it to the transcriptome to get evidence that this gene is indeed functional in stereocolon. We then did the same thing with Lepraria, pulled out REC8 and the flanking genes. First compared the flanking genes to stereocolon to make sure that they were indeed fungal and that we had pulled out a fungal contig from the metagenome assembly, which included REC8. We then compared start and stop codons in the gene model and looked for support for the gene model in the transcriptome of Lepraria, and found that for REC8 and for all of these other genes that the gene models matched. Lepraria seemed to have the same start and stop codons, and that the transcriptome indicated that these genes were likely functional. In addition, we did the conserved domain search again and found across all these genes that Lepraria and stereocolon have the same functional domain. So if stereocolon is using these genes for sex and they are functional, then Lepraria likely could use these genes for something like that as well, and that they remain functional. Again, taking a step back and looking at our phylogenetic approach, in general, across all of our samples of Lepraria, we found that all of these genes were complete or were intact and appeared functional. The dark blue indicates some partial gene models that were not due to like an incomplete premature stop codon, but were instead due to fragmentation of our short read assemblies. Interestingly, the only non-functional meiosis-specific gene that we found was non-functional across stereocolon. So interesting question for future research. And so stepping back again, we not only looked at the meiosis toolkit genes, but we also looked at a suite of 45 genes that have been shown to be involved in fungal reproductive structure formation. And again, same pattern across all these genes. They appeared intact and functional in Loparia as well as stereocolon. So it probably, moving back to our 3 lines of evidence, do we see loss of function in these sexual genes? No, we don't. They appear functional just like they do in the known sexual stereocolon. So yeah, so Loparia likely have the ability to make these structures and to do meiosis. So moving on to our final line of evidence, we asked about the pattern of selection across the phylogeny, comparing our test data set of Loparia sequences to a reference, here, our sexual stereocolon. We used a program called RELAX, which uses a modified branch site model to compare DNDS between the test data set and the reference data set. And it calculates a metric called K. And when K is less than 1, that's evidence of relaxed selection being more common in the test data set compared to the reference data set. And we found mixed results across the meiosis-specific genes and the rest of the genes that we looked at. A few showed intensification, a couple showed relaxation, but in terms of sets of genes that do specific functions in meiosis, there was no general pattern of relaxed selection across all these genes, indicating that that complex isn't functional. So moving back to our 3 lines of evidence again, our final question was, do we see in general relaxed selection on meiosis genes, or, and this is the same for the remaining genes in the toolkit and for the genes involved in apothecia formation, do we see relaxed selection? No, we don't in general see a consistent pattern of relaxed selection across the phylogeny. So if we pull back and think again about our initial question, do Loparia genomes in our bioinformatic analysis suggest that this genus has lost the ability to have sex? No, they don't. They appear to have everything intact and to be just as capable of having sex as the known sexual stereocolon genus. So then we have a new question. If, as lichenologists claim, for 200 years of super careful observation of Loparia, they have never been noted to form a sexual reproductive structure, what are they doing with these genes? Are they doing something else? Maybe they're very, very discreet at having traditional fungal sex, but if lichenologists are correct and they never form these structures, what are they doing with these genes? So I will leave you with a final hypothesis and a final question for future research. So based on things that have been found in other fungi, and some suggestions from the literature, maybe these Loparia are doing some sort of parasexual reproductive mode, or they have a parasexual reproductive mode. And this was suggested as one of the 27 possible types of reproduction in lichens by this Tripp and Lendemer paper, where they showed that there was some evidence of hyphal fusion between some Loparia lichens. And it's possible from our data, maybe we can add to this, that maybe there is a fusion and some sort of mitotic recombination that actually has co-opted these mating and meiosis genes to do something that is adjacent to sex. So interesting future work to show recombination and function of mating type genes in this supposedly asexual, but maybe not, system. So with that, thank you to all of my co-authors, Alejandrina, Felix, Torsten, and Yukon, and to Todd and the Pritzker Lab at the Field Museum, and to our Russian UIUC genomics course that helped us with all of our sequencing. Thank you.
Hello. Thank you all the speakers for great talks. And with this, I would like to open for questions. All right, so the first one is for Arif. Chris Keeling thinks that it was a very nice presentation. It was. I'm curious about the Y chromosome also. How is it conserved within Coleoptera?
Thanks very much. Yes, so I'm also curious about them. So among the 180-ish genomes that we have in possession, probably less than half of them has the Y chromosome assembled. But usually, as you probably know, they are very tiny compared to the X and also the rest of the chromosome. So that's why they're called YP or Y parachute. So probably contain very few genes. And since my analysis was Busco-based analysis, and we found almost no Busco genes for almost all of the species. So at the moment, I don't know what to expect with how about the conservation of this Y chromosome, but maybe we can try other approaches, maybe synteny analysis, or that doesn't require Busco, because Coleoptera doesn't have any Busco for Coleoptera. We use Endopterygota. Meanwhile, for Lepidoptera, they have their own Busco. Yeah, so that's my answer.
All right. Thank you, Arif. The next question is from Meredith. Mark Blackster is asking, there was one clade of, I will not even attempt to pronounce the clade of the sexual lichen, that had a significantly longer branch length than the others analyzed. Do these 2-3 taxa bias the limit of your ability to detect relaxed selection overall?
That is a really good question. You should have been a reviewer on my paper, because then you would have made me redo all of the analyses, dropping those 3 taxa, which I did not do. And in retrospect, I probably should have. But paper just came out in BMC Genomics, so it's a little late to try that. But yes, it could maybe, but I didn't actually test that, and I should have. I'm sorry.
I also had a question about for you. I wanted to ask whether you could just look at the more classical consequences of sexual reproduction, which is recombination, and just if you look at the population structure, you can just find an allelic combinations that are impossible to see without sex.
Yeah, that's the next lot. That would be the next logical step, I think, in this study. We write about it in our discussion, that collecting population level data from some of these Liparias and looking for evidence of recombination using kind of classical population genetics techniques would be the next step. We know there's another group working on this question, and I think that they have all of those data. So stay tuned for stuff coming out of their lab. I think that from what I've heard from an abstract of theirs that came out, that some populations do show evidence of recombination, and like the occasional population they find doesn't. But that paper's not out yet. This is just like an abstract I read that they had published. So hopefully those data are coming, but not from us, unfortunately.
Oh, great. In this regard, they would be exactly the same as the rotifers, right? They also see what is compatible with. Okay, thank you. Do we have another question? There is a question for Chloe, which I will send her. And no, I don't. Oh my gosh, we run out of questions. Or am I blind?
There's one about tag boundaries.
Kerstin asked, Arif, we heard yesterday that chromosome fission points in Lepidoptera are often associated with tad boundaries. Is this something you observe in beetles as well?
Very nice question. I actually haven't heard or read about the paper yet, so I don't have any prior expectation whether it's also the case for beetles. But that sounds like an interesting idea to pursue, maybe. Thank you.
Yeah, I'm just really bad at scrolling. So Now is one from Philippe for Meredith. Sorry, but could you relate these genes with the sex? I'm not sure what are those genes.
I don't understand.
Yeah. All right.
I mean, the 8 genes that I focused on in the talk were genes that in other organisms are kind of like the classic meiosis genes that you look for that, at least as far as research knows so far, don't have functions outside of meiosis. So if they're functional in an organism, they're probably doing something meiosis or meiosis-like, some sort of recombination. There are a couple of papers that in non-lichen systems, in I think like maybe some protist systems. I can't remember the exact organism, that also hypothesize that some of these genes might be used for like a mitotic recombination in a parasexual type of reproduction. But I don't know if there yet is much information on like expression during parasexual reproduction. And there may be in some model fungi that I don't know.
Great answer for a very vague question.
That's the best I can do.
Mark is asking about what is the pattern of heterozygous state among Gleparia genomes? Are there signals of recombinations? Oh,
yeah, this is similar to your question,
Yeah.
I think. So we don't have population level data
but
for any of these species. Everything in the phylogeny, almost everything in the phylogeny was a single species. So maybe we could have looked at this.
Was there ploidy?
No, they're haploid.
They're haploid. I see.
They're haploid. Yeah. Yeah. So you can't look at like Hardy-Weinberg or anything like that. And you can't look at heterozygosity within a genome. So you'd really need population level data to really get at it. I mean, I guess you could kind of do a cross species, but I don't know.
So asexuality in diploids can be also that they still recombine, but only within the 2 haplotypes they already have. I think that having a haploid system is a really cheeky way how to solve the problem. I like it.
It kind of broke my brain when I started to work on it because I've worked on diploids forever. And when I started to work on lichens, I was like, oh, this is a whole different way of thinking of things.
So there is another one by Dominic Groffman who says that the really fascinating talk, Meredith. Thank you. It's fascinating that this diverse genus is entirely asexual. Would parasexuality explain the diversification in
Lepraria. Yeah, that's a good question. I'm not 100% sure, but I know that there are some modeling papers, at least for systems where they're mostly asexual, but sexual reproduction occurs occasionally and very rarely. And I know that the models say it's something like I can't remember. Like once every 10 generations or something is all that they have to have sex in order to like maintain the like genome integrity and maintain the variation necessary for adaptation and diversification. But I'm not sure how that would translate to parasexual reproduction, like how common parasexual reproduction would have to be. But I think my guess is that it's a lot less common than you would infer that it would have to be, at least from the PopGen models. Mm-hmm. So probably it could.
So I don't know if you've seen the mite papers that came out recently that are showing that they have like a genetic evidence that they really do mitotic asexuality. Anyway, there are evidences of asexual species that actually do have more ethic and purifying selection, which is somewhat great. Anyway, thank you everyone for a great session. It's been lovely hearing your talks and chatting to you, and I will encourage again the audience to join the Tree of Sex Consortium. See you next time.